Okay, hello people. We are finally away from proteins and enzymes. We're going to talk about lipids, another major macromolecule in the biomolecular world. Okay, so after watching this video, you should be able to define key vocab terms. It only helps if I actually pick a color. And there's going to be a lot of key vocab terms. Draw fatty acid from symbolic notation and vice versa. This means that you can get from symbolic from the structure to symbolic. Draw a tag from symbolic notation. Recognize phospholipids and glycolipids. Compare the functions of energy storage and membrane lipids based on their structure. So again, structure implies function and vice versa. So we're going to talk a lot about how the structure of the different lipids influences their function. Predict melting temperature properties based on fatty acid structure, and then describe the formation of a lipid bilayer. This should be review. Okay, so we're going to have three days essentially of lipids, and this is going to be lipid structure function day 1. So we're going to do lipids that are used as fuel storage or energy storage first, and then we're going to do lipids that are used in membranes, and then the next day we're going to do lipids that are involved in signaling. So lipids have a lot of different functions, and hopefully you'll see that that's because there are a variety of different structures that are all considered lipids. So what are the structural requirements of a lipid? So oftentimes we have this weird cartoon where you've got this polar head group and a non-polar tail. And in biochemistry, we like to do a little bit better than just a cartoon representation. So on this, I've got a number of different representations of lipids. So that polar head group is something that would interact with water. So it would form a lot of non-covalent interactions. We would have an ion dipole here, another ion dipole here, ion dipole, and these are all with water. So all of the head groups, the functional groups at the top part of the structure are polar, and that's why it's called a polar head group. And then the tail is a nonpolar tail, and you can see that it's all CH bonds and CC bonds, which we know are nonpolar. So those are the requirements of being a lipid. Lipid has to have a polar head group and a nonpolar tail, and this can come in many different formats. For example, there's this one little polar head group here on one side of the molecule, but the rest of this is nonpolar, so that makes this a lipid. You can see over here, what's in blue here is very polar, lots of oxygens, and then what's down here is nonpolar. So you have a region of the molecule that is polar, which is separate from a region of the molecule that is nonpolar, and of course this means that all of the lipids are amphipathic. Also amphoteric, and all of the different words that you have for something that has both polar functional groups and nonpolar functional groups on the same molecule. Okay, so we're going to start with the fatty acids, and we're going to build our way up to something a little bit bigger. So fatty acids are exactly how they sound. There is a fat region, or a nonpolar region, and then there's an acid. So hopefully you recognize this as the carboxylic acid functional group. Fatty acid, fatty acid, fatty acid. Do you see why we call it a fatty acid? So the nonpolar region with the CHs and the carbon-carbon bonds, they sometimes call this the greasy part of the molecule. Food for thought. Okay, the carboxylic acid has a pKa of approximately 4. So at pH 7, the carboxylic acid should have a negative charge on it. So it should be a -1 and be charged. Food for thought. Most fatty acids in the body are between 12 and 24 carbons long, and they are usually even numbers in length. So the number of carbons is 12, 14, 16, 18, 20, 22, 24. Those are the typical lengths. It is possible to have odd number fatty acids, but they're a little bit more rare. Okay, let's do some vocab. All right, so these are three, I'll say, common fatty acids. Stearate, also known as stearic acid, is considered saturated. F.A is my abbreviation for fatty acid, and saturated just means there are no double bonds. I should have wrote that one out. Then maybe there's going to be too many acronyms. No double bonds. Okay, so saturated means that every carbon has every possible hydrogen it can, except for, of course, the carboxylic acid. Oleate or oleic acid is unsaturated. This means it has at least one double bond. That's what unsaturated means. It has lost two, in this case we've lost two hydrogens to get to that double bond, so it's unsaturated, missing some hydrogens. Linolenate or linolenic acid, which is my favorite fatty acid just because it's super fun to say, has a couple double bonds here, so we would call it poly, meaning many. Whoops, I just about wrote many. Go back. Poly unsaturated fatty acid. Poly, again, means many double, in this case would mean many double bonds, more than one double bond. Okay, and then with the double bonds in fatty acids can be either cis or trans. Most double bonds in dietary fats, these are fats that you eat, that's what dietary means. Fats or fatty acids are cis, and there is a reason for that, and we'll get to it later. But here, this is transoleic acid and cisoleic acid, so again, you can see that the trans double bond, if you remember your torsion angles, means that this will have a 0-degree torsion angle. And the cis double bond puts the functional groups on the same side. That means this R group and this R group are on the same side. This would be an R group here, and this would be an R group here. And the hydrogens that are here are also on the same side. That's what cis means. It should have a kink in the bond. And trans, they are on opposite sides. So R group, R group. 1, 2, 3, 4, yeah. And the hydrogens are also on opposite sides. Okay, food for thought. All right, so now we're going to get into symbolic notation of fatty acids and how you draw a fatty acid based on symbolic notation. All right, so fatty acids have this really interesting nomenclature that we're going to go through. So it's called symbolic. Notation. And the first example we're going to do is 20 colon, let's do 2 parentheses, trans delta, let's do 14 and 16. Okay, so the first number tells you the total number of carbons. The second number tells you the number of double bonds. Okay, this tells you the shape of the double bond. And then this delta with the numbers tells you the location of the double bond. Okay, so let's draw this one out. So I always start with the carboxylic acid, OOO minus, and if you remember, hopefully from organic chemistry, this carbon is going to be the highest priority, so we call it carbon number 1, and the reason it's highest priority is because it has the most electronegative atoms attached to it, which makes this one number 2, and then I'm going to keep going. This will be 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. This is carbon 14. And you'll notice that we'll have a double bond on carbon 14, and it's trans. So I'm just going to do this. 14. I drew way too big. It's okay. 14 This is carbon 15, and then 16 is here. Another double bond starting at 16, going to 17, 18, 19, and 20. Okay, so that's symbolic notation. Let's just do one more for practice. I'll do it in a different, let's do 18:3 cis delta 7, 9, and 12. All right, so we have 18 carbons total, 3 double bonds, and the double bonds are at carbon 7, 9, and 12, and they're all in the cis shape. So again, COO minus. Carbon number 1 is this one right here. So 1, 2, 3, 4, 5, 6, 7, and 7 has to be cis. That makes this one 8. This is 9 to 10, 11, 12, and 13, 14, 15, 16, 17, 18. Okay, my recommendation is that you add the numbers to every single carbon just to make sure, like just triple check that you actually have the right number of carbons and you didn't forget. See right here, I added, this should have been 18 up here, so checking my work means that I do that. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Okay, cool, looks good. All right, I gotta go back. All right, so next we're gonna talk a little bit about mixtures and like what happens when you put a whole bunch of fatty acids in a really small space. So the first thing we're going to talk, I'm going to do groups of these. So I'm going to have a mixture of fatty acids first that are all, oh, that looks terrible. Yikes. Saturated. Sorry, those should all just be regular zigzags. All right. Okay, then I'm going to have one that has some trans double bonds in it. So we can talk a little bit about melting temperature. And notice that my trans double bonds are not always on the same double bond. That part isn't essential. Okay, and then of course I don't leave myself enough room. Big surprise. You go over there. And then my third mixture, or my third grouping, is going to be cis. And it turns out that the cis double bonds cause kinking, in the structure. I'm just making these up as I go. Hopefully you get the idea. I'm only going to draw 2 of those. You can imagine that there's like this whole population. Okay, so it turns out that if you put a whole bunch of saturated fatty acids together, you get something that's the consistency of butter, or you'll get a solid. And that's because between all of these chains here, there's a ton of van der Waals interactions going on, which makes it very, very stable. Also, you have really good shape interaction, so these stack together really, really nicely in this nice, I don't want to call it pancake fashion, but kind of puzzle piece fashion. And because of that, the melting point for a mixture of saturated fatty acids is actually pretty high. Hopefully that makes sense. And then when you move to trans, you lose some of the van der Waals. You still have a whole bunch of them. You lose some of the van der Waals, but what this ends up being is more like spreadable butter. Okay, so you still have lots of van der Waals, there's just less of them, and you also have good shape interaction. So it's still stable, but less so. But less than our butter. It's called butter. Okay, and then when you pack a whole bunch of cis fats together, or the ones that have cis double bonds, you get something that's a consistency of like olive oil or the vegetable oils. And what's happening is because of the kinks here, they're just too far away to make all the van der Waals interactions. So this is the least amount of van der Waals, and the shape interactions are very bad. And by shape, again, I mean like they don't fit together like puzzle pieces. And the melting temperature on these is the lowest of them all. We'll call this strongest or most stable to weakest. And then this is, strongest, most stable to weakest, going in this direction. Okay, so these are mixtures of what might be considered pure fats, where like these are all saturated, these are all trans fats, and these are all cis fats. Okay, and if we look at a different diagram of this, so again, here's our saturated fats, and here's a mixture of saturated and unsaturated, and it turns out we can kind of classify or rank the interactions based on melting temperatures. So in general, saturated fatty acids have a greater melting temperature than unsaturated. And trans, in general, have a greater melting temperature than cis fats. And then a pure fatty acid mixture is greater in melting than a mixed. And for example, this is a mixed or a mixture. This is all, of course, assuming equal CH tail length. If the tail length is not assumed to be the same, then the longer the tail, the higher the melting temperature. Okay. So a couple slides here on typical fats in a human diet. Lard is pig fat, which is animal fat, which hopefully you can see is a solid. Ugh, gross. You would also predict that because it's a solid, it's made out of mostly saturated fats, because saturated fats packed together, lots of van der Waals, the shape interaction is great, super stable, so it forms a solid at room temperature. Whereas something like vegetable oil doesn't form a solid at room temperature, and the most abundant oils are oleic acid, linoleic acid, alpha-linolenic acid, and these are 60% of this is all cis fats, and 7% is saturated. And I'm going to give a little shout out about canola oil, because most oils are named for where you get the oil. For example, olive oil comes from olives. Peanut oil comes from peanuts. Sesame seed oil comes from sesames. But canola oil comes from this plant that they grow a lot of in Canada called Brassica rapum. And it's a source of what's called rapeseed oil. Traditional plant breeding was used to create a seed that is rich in vegetable oil. And rapeseed oil is not marketed as rapeseed oil is not marketed as rapeseed oil because no one would buy it. And instead they call it canola oil, and the name comes from the place where it's grown the most, which is Canada. Food for thought. Literally, food for thought. Weird. And then we're going to talk about these omega-3 fatty acids, or omega-369s. Canola oil contains a lot of omega-3 fatty acids. Alpha-linolenic acid is also considered an omega-3 fatty acid. And the omega-3 Means that the first double bond is on the third carbon from the end of the chain, blah, blah, blah. Who knows what that means? Let's draw a picture. Doo-doo, 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 doo-doo. Okay, 1, 2, 3. So the way that this is named is that the beginning of the chain is the alpha, and the end of the chain is the omega. So the beginning and the end, the alpha and the omega, these are the beginning letters and the last letters of the Greek alphabet. Now, if you omega fatty acids, you count from the back, or the omega, and you go 1, 2, 3. If the double bond occurs at the third carbon, it's an omega 3 fatty acid. And when you draw these, make sure that you always draw them as cis, because omega-3 fatty acids are healthy for you. And the reason they're healthy for you is because they don't stack nicely and they don't form butter. So what happens is you eat this, okay, and some of the fat that you eat becomes incorporated into the lipids of your membrane. Okay, so just imagine these are all the lipids. You want this membrane as fluid as possible. And the way to get that is by incorporating cis fats into fatty acids into the membrane. If you incorporate too many saturated fats, then you basically get what I'm going to call the butter cell membrane. The membrane becomes solid, and then nothing passes through. Solids tend to be a bad thing in your body. We tend not to want to have solids. Think about like kidney stones and calcium deposits and all sorts of things that end up being solid in your body. You don't want that. Anyway, so when you draw your omega fatty acids, make sure that you always draw them cis, because omega-3 fatty acids are good for you when they get incorporated into your membranes. They help keep your membranes fluid so that things like oxygen freely flow across. Oxygen would not be able to flow across the butter cell. I know it's kind of gross. And you want CO2 being able to come back out of the membrane, and this is all made possible by a fluid membrane, and a solid membrane is bad. Mad. Okay. Cool. So there are many food applications where it's desirable to have the texture of a solid fat with the inexpensiveness of vegetable oil. So vegetable oil is super cheap. And if you hydrogenate vegetable oil, you essentially get the texture of the creamy filling in an Oreo cookie. Okay, so, oops, come back here. So what happens is in the vegetable oil, canola oil, whatever, there's a whole bunch of these cis fatty acids, okay? And if you hydrogenate them, which means H2 and a catalyst, palladium, you'll add hydrogens across all of these double bonds, and then they no longer become double bonds, they become single bonds. Not all of them. So you don't get complete hydrogenation, you get partial hydrogenation. And that partial hydrogenation gives you just enough to get it to be kind of sticky, but not solid. Okay, sorry about that. So yeah, so partial hydrogenation will give you results, or gives a mixture of cis fats, trans fats, some saturated fats, and the result is you get that texture right there. And it's super, super cheap to go from vegetable oil to, I don't even know what you call that. It's still a fat. Gooey, fluffy fat? I don't know. Okay. And, well, I gotta pause for a second. All right, so fatty acids used for fuel storage versus cellular structure. So it turns out your free fatty acids are just not floating around your body. Or in the cell. They're tied up, and they're tied up in either a tag, which is the abbreviation is tri, 3, acyl, this is the nonpolar tail, and glycerol. They're tied up in tags, or they're tied up in phospholipids. And the very basic structure is 3 carbons in a row, CH2, CH, CH2, and then an ester, or actually I'm gonna actually show you what the R is. The R is the acyl tail, or what was the fatty acid tail. This is also OCOO. Okay, so that's the basic structure of either a tag or a phospholipid, but what differentiates the two is whatever is attached to that, if you call this the first or the third carbon. And we're going to talk about the first one, tags, and then we'll talk about phospholipids second. Okay, so triacylglycerols occur when you esterify 3 fatty acids onto a glycerol. All right, get ready for a mechanism. I know you're super excited about this. Okay, so here's the structure of glycerol. You should have this structure memorized. Please memorize this. Please memorize this. It's a very important molecule in your body. So it's 3 carbons, and each of them gets an OH. Okay, this is glycerol. And then I'll put my fatty acid, different color. Okay, so I'm going to show you the mechanism of esterification just one time, and I'm going to do it kind of fast. So it's actually this OH that's the nucleophile, okay? And then, so it has a partial negative on it, whereas this carbonyl carbon has a partial positive, and everything in chemistry basically boils down to opposite charges attract. So this nucleophilic atom is going to attack this electrophilic atom. There will be, well, there's always water around here. So we gotta make a good leaving group. Electrons will go up, they'll come back down, and what's get kicked off is this O, which could be an OH. I could grab this one to make it a water. So H2O leaves, and the resulting first round is CH2, CH, CH2. These haven't been esterified yet. Is the red oxygen is still attached to glycerol, which is now attached to a carbonyl carbon, which is attached to the acyl tail. And then this will happen 2 more times to get the triacylglycerol, which is this molecule right here that's represented in skeletal structure, and then it's represented in spheres. Okay, so what? Who cares? What difference does this make? Why is the tag, or triacylglycerol, such a great fuel storage or energy storage molecule? Okay, so what is it about this particular structure that makes it so great as an energy storage molecule? Like, why don't we just keep them as fatty acids? What's the big deal? So there's a few things that happen. First of all, you'll notice that the fatty acids all have this negative charge on them. And if you were to put a whole bunch of fatty acids in a teeny tiny spot, those negative charges would begin to repel off of each other. Okay? So when you tie up that negative charge and you get rid of it and you add it to the glycerol backbone, there's no more repulsion. So you can get even more fatty acids in an even tinier space. Okay? The other thing that's really awesome about the tag is that in general, the more CH bonds, the more energy is stored. The more energy is stored. And so a tag is able to store a lot of CH bonds. I mean, this is basically all CH bonds. Is able to store a lot of CH bonds in a teeny tiny space. Okay, so those are the things that make tags such a great fuel storage, is you can get a lot of CH bonds in a tiny space and you alleviate the repulsion of the negative charge of the fatty acids. Cool, huh? Okay, so moving on to phospholipids. Phospholipids are usually the ones where you've got the head and then it's got two tails. If I was to represent a tag, I would probably represent it as a head and three tails. Tag. But the phospholipids are usually always represented by two tails, and the reason for that is that that third, what would have been the third tail, is no longer an acyl tail. So these R's represent the tail. So there's two tails here. That's the two squigglies. The third, what would have been the third one, is actually a whole different functional group that makes it really great for membranes, structural support and membranes. And these are all called phospholipids because they all have phosphates in them. Okay, these are the 4 phospholipids that you need to know. You need to be able to recognize them and draw them. So we're going to talk about each one of them separately so that hopefully you'll be able to see the different structures. And if you'll notice, these are all still attached to what would be called the glycerol backbone. So we only had 2 rounds of esterification. So these were once the fatty acid tails. Okay, so the first thing that you'll know, this is called phosphatidylcholine. This functional group with the NH3 or the nitrogen and 3 methyl groups is the choline. Okay, so phosphatidylcholine means it has a phosphate and a choline on it. Phosphatidylethanolamine. This is ethanol with the 2 carbons, and this is an amine, so it's ethanolamine. This one down here, whoa, hey man, you're not the eraser. Okay, green. This one down, let's do this one. This one is phosphatidylserine. I should have just erased that. Phosphatidylserine. So this is the backbone, NCC, and then the side chain is going this way, so CH2OH. That's phosphatidylserine. And then phosphatidylglycerol. Here's the glycerol, carbon 1, carbon 2, carbon 3, and they all have what was an OH. And then the other type of membrane, or another type, not the other type, another type of membrane lipid are called sphingolipids. And the sphingolipids have this really weird functional group where there's no more ester. Instead, there's this funky OH double bond situation going on here. You only need to be able to recognize recognize, not draw these. They have a fatty acid chain, but it's attached to an amide instead of an ester. And then this X could be anything, and I just wanted to give you an example of a membrane lipid that's attached to sugars, because we call these glycolipids. Okay, these are all sugars. The reason that you know that they're sugars is because there's a ring with lots of OHs. And the prefix for anything sugar related is usually glyc or glyc with like an I. See, yeah, so glyc, glyc, gluc is another one. But you're looking for these rings with a lot of OHs coming off of them. That's how you know something is a sugar. We would also call these carbohydrates. Lots of words that just mean the exact same thing. So that's a glycolipid. All right, I really like this slide. I think it's a really cool summary slide. So you have your storage lipids or your energy lipids, where it's a glycerol backbone and then 3 fatty acid tails. And then all these other ones over here, they're membrane lipids. Again, glycerol backbone, 2 tails, 2 fatty acid tails. That's where, again, this funky cartoon structure with the 2 legs comes from. And then anything else coming off of it is the polar piece. This one is phosphatidylcholine, or not quite phosphatidylcholine because it's got the sphingosine connection in it. Okay, and just as a quick reminder, you have actually seen this figure before, so I'm hoping that you'll be able to describe the formation of a bilayer, a phospholipid bilayer, based on the hydrophobic effect. Okay, and then maybe also ask yourself, why don't we use a tag for lipid bilayer? Why is that not happening? Okay. There's a couple animations that I uploaded to our Canvas site that I hope that you have a chance to take a look at, because for one, it really shows the fluidity. They really show the fluidity of phospholipids in the membrane. Okay, and phospholipids have a couple different ways that they travel through the membrane. They can travel left and right, so within a layer, or they can do this funky flip-flop thing, and this is really slow, and it sometimes requires a catalyst in order to do it. But the reason that you may want to flip-flop membranes like this is that these polar head groups can be used for signaling purposes. So if you want whatever signal this might carry with it, if you need it on the outside of the cell, then the cell will flip-flop that phospholipid. Cool, huh? Okay, and then this one is the last membrane lipid, is the sterol. Sterol, because they have this alcohol group on them. They're still considered lipids because they have a nonpolar part and a polar head group. Another thing that you should notice about lipids is that they have this steroid nucleus. And the steroid nucleus is just a unique functional group feature of anything that has this specific ring structure. So it's 3 6-membered rings and then 1 5-membered rings, and they're always connected in this fashion, where the first two are right next to each other, and then the third one is up high, and then the last one is over to the side. And then they may have many different functional groups coming off of them. Again, this is a steroid nucleus, so you're going to see this when we talk about steroids, but it's common, and it is what do I want to say? I don't want to call it nomenclature, but Symbols for it are A, B, C, and D, going in this case from left to right. So what is it that sterols even do for the membrane? So once a lipid bilayer has formed, it's not just phospholipids. So here's a phospholipid. The membrane is made up of a lot of more and unique different things. We're going to talk about proteins that get added to membranes, sugars that get added to membranes. But sterols slide into the membrane, and you'll see there's one per side here, where the polar head group is on the outside, hanging out with water, just like all of these. The phospholipid tails, or I'm sorry, the phospholipid head groups are also hanging out with water on both sides here. And the sterols have a function at different temperatures. So at high temperatures, the sterols add rigidity. So the membrane doesn't fall apart. Okay, and at low temperatures, the sterols increase the fluidity by keeping, no, I'll say this, by also binding it, wow, rough day, binding in between the phospholipids I ran out of room. By binding in between the phospholipids and keeping them from sticking together. Okay, so different functions at different temperatures. So at high temperatures, it increases the rigidity and keeps the membrane from being too fluid and falling apart. And at low temperatures, it increases the fluidity. It keeps it from being too solid by keeping the phospholipids from sticking together. And this is just my last sort of shout out slide. We talk a lot about the cell, and sometimes I'm not sure that you have an idea of what that looks like. So I might recommend that you kind of review eukaryotic cell structure. Probably should have done this at the beginning of the semester. I'll know better next time. Eukaryotic literally means true nucleus, and this is an electromicrograph of biological membranes. So there's a number of organelles that have membranes. There's the nuclear membrane that surrounds the nucleus. There's all these waves that are around the nucleus. That's the endoplasmic reticulum. Not around that. I didn't want to highlight that. Around the mitochondria, here you are, mitochondria, there's a whole bunch of membranes, and then of course there's the outermost part of the cell that is the cytoplasmic membrane or plasma membrane. There's many different ways of saying cytosol. Plasma is another way. And then this is just to give you an idea of the thickness of the lipid bilayer that surrounds all of these organelles and separates them from the environment, or keeps things inside, keeps things outside. The total distance is 30 to 40 angstroms. These little squiggles in here are your cholesterol. And in a few days from now, we're going to talk about how we can add, or what the function of adding proteins and sugars to the membrane are. And I know that was kind of a long one. But hopefully you're able to keep up, and we'll see you in class. Bye-bye.